Hi everyone, today a video on biological chemistry and specifically enzymes, DNA and anti-cancer drugs. So enzymes are proteins. They're made up of the proteins that we looked at in the previous video, the idea that essentially they are built up from amino acids as well. And so they act as catalysts in living organisms. And enzymes are extremely specific. Each enzyme is optimized for a single reaction, for a specific purpose. Now, enzymes are what are called usually globular proteins, just a specific type of protein, and their shape has a cleft or a crevice in it, essentially a part of it where it kind of juts in, and this is called the active site. This is where the reaction takes place. And so the reacting molecule, the substrate, will fit precisely into the active site of a specific enzyme, and it will be held in the correct orientation to be able to react. Now, only molecules that have the correct 3D shape will fit the 3D shape of the active site and can react. It's incredibly shape specific. And this is often referred to as the lock and key mechanism, the idea that only a specific key will work in a specific lock. The same way here, in that only a specific substrate will be able to fit perfectly into a specific active site of an enzyme. Now, in reality, the true picture is a bit more complicated because the reacting molecule, the substrate, must not only fit the shape of the active site, but it must bond to it temporarily by intermolecular forces, non-covalent intermolecular forces. And now, while it's bonded, these forces will promote the movement of electrons within the substrate. They essentially hold it in the correct orientation and hold it there for it to be able to react. And in doing this, it lowers the activation energy of the reaction. It acts as a catalyst. It gives the reaction an alternative pathway to proceed down, one that has a lower activation energy and therefore can occur more quickly. And so generally we have our enzyme with our active site, we have our substrate or sometimes substrates that will bind to our active site using these intermolecular forces, these non-covalent interactions. They're held in place as the reaction takes place, and then they leave. They sort of unbind from the enzyme. They produce our product, and our enzyme is left unchanged. Remember, it's a catalyst, so it's unchanged, and its active site is now free to be able to go and catalyze the same reaction again. So we talked about how shape-specific the active site of enzymes are. And what this kind of leads to is that many enzymes only catalyze reactions with one molecule of a pair of enantiomers. Remember that enantiomers are stereoisomers. They're kind of like your hands. They are non-superimposable mirror images of each other. And so these enzymes are said to be stereospecific. They only allow one enantiomer to come in. And so the cartoon at the top right here kind of shows that. We have these different coloured balls, a triangle and a rectangle here, trying to fit into the certain specific holes here. And you can see that the one enantiomer, the R enantiomer, fits perfectly in, the shapes fit, but then the S enantiomer, for example, you're trying to fit this rectangle into this circular hole, and so it just doesn't work. And so because they're so shape selective, they only allow one enantiomer in. Now, drugs can also be designed that will affect the action of enzymes. So, for example, if we have an enzyme that is catalyzing a reaction that we don't want to happen in the body, maybe it's producing a harmful substance, or it's allowing, for example, cancer cells to be able to continue to sort of grow and divide. If we can block an enzyme from catalyzing a reaction, it stops the reaction occurring. So in order to block the action of the enzyme, we need to have a drug molecule or an inhibitor that will bind to the active site, something that looks like the substrate, but that will bind to the active site and stop the actual substrate from binding to the active site and getting catalyzed. Now, enzymes can also be easily denatured, so they can lose their 3D shape and have it changed. We talk about the fact that the secondary structure and tertiary structure of proteins can be easily disrupted and the shape changed. Because enzymes are made from proteins, the same thing happens here. We can easily change their shape simply by small changes in temperature or pH. Now, inside the body, drugs can be introduced that mimic the natural substrate of an enzyme. So it's something that looks like the natural substrate. It will bind to the active site rather than the actual substrate and block it. It won't allow the actual substrate to come in. And this is something that we call enzyme inhibition. And technically, this is what we call competitive inhibition. There's another type called non-competitive inhibition, where we have our drug molecule, our inhibitor, binding not to the actual active site of the enzyme, but to somewhere else on the enzyme. The actual active site of an enzyme is a very small part of the whole enzyme structure. But by binding to the enzyme, because this is a very specific 3D shape, by binding to somewhere else on the enzyme, it can change the active site. It can change the shape of the active site, meaning the substrate then can't actually bind in itself. And again, we stop that reaction from occurring. So chemists can now use computer modelling packages to be able to predict the shape of proteins before they've even been synthesized, simply based off this primary structure of amino acids and knowing how these are likely to fold into alpha helices, beta sheets, and the kind of intermolecular interactions we see that will hold them together. We can start to predict what the actual overall sort of 3D shape of a protein will be, and therefore the properties of the protein can be predicted, and this can help inform drug design. Now we move on to look at DNA. So DNA stands for deoxyribonucleic acid, and it's present in all cells, and it contains the blueprint, the plan from which all organisms are made. So every living thing has its own particular DNA molecules, and there's an infinite variety of DNA. And a single strand of DNA is a polymer, and the polymer is made up from just 4 different monomers. But it's the way that these 4 different monomers are arranged that leads to this infinite variety of products. And so the monomers that DNA is made from are called nucleotides. And a nucleotide itself is made up of 3 parts: a phosphate group, a sugar, and a nitrogen base. And these are all bonded together, as you can see at the top. Now there are 4 different bases that are called cytosine, thymine, adenine, and guanine. And these have a 1-letter code, C, T, A, or G. And the structures of them are shown here at the bottom. And you can see that generally adenine and guanine look kind of similar. Cytosine and thymine again look sort of similar. And generally they contain quite a lot of nitrogen atoms. So much like how we saw condensation polymerization being used to make amino acids into peptides and proteins by forming peptide bonds or amide bonds, 2 nucleotides can link together when the hydroxyl group, the OH group, on the phosphate of one reacts with the hydroxyl group on the sugar of another nucleotide. So this is again another condensation polymerization because we're going to eliminate water. And essentially it's the OH group on a phosphate of one and the OH group on the sugar of another coming together to make this kind of phosphate link and eliminating water. And again, much like we saw with the amino acids, once we did this to make a peptide, we still had a free amine end and a free carboxylic acid end. Here, when we do this, we still have a free sugar OH group and a free phosphate OH group. So this can continue to go on and form much longer polymer chains. And so eventually this forms a really long chain which consists of a sugar-phosphate backbone, and the bases that are attached protrude outwards. So you can see here that we've got this nitrogen base that's attached onto the sugar, and you can see that it kind of protrudes to the side of it. And the arrangement of these bases along this chain can be in any order, hence the infinite variety of DNA molecules. Now, DNA molecules are specified by the ordering of the bases. So much like we had our proteins being specified by their primary structure, the sequence of amino acids, DNA is specified by the ordering of the bases. And again, we can talk about a 1-letter code. So it could be, for example, the 1-letter code shown here. Remember that we have G, C, T, and A. Now, cells in the bodies of all living organisms can read this code, and this code contains the instructions, the plan for making living things. It's the recipe, if you like, for making proteins. And of course, proteins, as we said in the previous video, are the basis of all living things. Now, chemists can also read this code, and this is something called DNA sequencing. So we can actually, synthetically in a lab, make DNA. So DNA exists as a 3D structure as well, and the 3D structure that it exists as is this called the double helix. And so it exists as 2 of these strands, these strands that contain the sugar phosphate backbones, these polymer chains, with the bases protruding outwards. And so 2 of these are held together by hydrogen bonding and pi-pi stacking. Now the bases that we saw, the thymine, the adenine, cytosine, and the guanine, can interact in these 2 ways. The first is hydrogen bonding, and I've shown that there at the top right. All of these, in the case of thymine, each of these thymine, adenine, cytosine or guanine bases have regions on them that they can either have hydrogen bond donor properties or hydrogen bond acceptor properties. The other thing that can hold them together is pi-pi stacking. So these bases are often aromatic. They have delocalized pi systems. And so these pi systems can then start to interact with one another, and kind of layering them on top of each other can introduce another intermolecular force. Now, there's only very specific hydrogen bonding pairs. So adenine and thymine can hydrogen bond together, A and T bond together, and cytosine and guanine bond together, C and G. And it's only these specific pairs that can hydrogen bond together effectively, and they're the only pairs that are seen in DNA. So A and T goes together, C and G goes together. So two DNA molecules can bond together if their bases are in a complementary order. For example, a C matching a G or an A matching a T. So if the first strand was G, C, A, T, G, C, T, A, A, then the complementary strand, all we need to write down is just the opposite of that, which one's going to bond to it, hydrogen bond to it. Remembering that a C matches a G and an A matches a T. So the complementary strand would be CGTA, CGATT. Now, if you look at the diagram that shows the hydrogen bonding, you'll see that the A and T pair has 2 hydrogen bonds compared to the C and G pair having 3 hydrogen bonds. Now, what this means is that the C and G pair, to break apart, if you wanted to kind of break apart the DNA strands, it takes more energy to break the C and G pairs apart than it does to break the T and A pairs apart, because there's 3 hydrogen bonds, not 2, so it's much stronger intermolecular forces going on in the C and G pair compared to the T and A pair. And so in order for the strands to neatly fit together, the 2 strands kind of wind around each other in the shape of a double helix. So on the outside of this double helix, kind of curving around, we have our sugar phosphate backbone, that polymer backbone. And then in the middle, because our bases from that, remember, kind of protrude outwards, our nitrogen bases. And so these bases then pair up A and T, G and C to kind of link the 2 strands together. And then finally, looking at anti-cancer drugs. So cancer is a group of diseases. And the defining feature of all of these diseases is that we have rogue cells. These are cells that have lost control over the growth and replication. And so these cells grow and divide much, much more quickly than normal healthy cells do. And they're able to essentially survive on their own. They don't need to use the blood supply of the body. They can create their own blood supply. They're able to evade the immune system much more easily. They kind of hide away from being able to be repaired or destroyed by the immune system. Now, cisplatin is one of the most successful cancer treatments, despite it being discovered many, many years ago. As a cisplatin, as the name suggests, is a square planar molecule based on platinum, where we have 2 chlorine groups and 2 NH3 groups. And both of those are cis, respectively, cis meaning that they are at 90 degrees to each other. Now, how it works, the mechanism of action, is it binds to strands of DNA, and when it does this, it massively distorts the structure of DNA. The perfect double helix is kind of disrupted, and it makes DNA essentially kind of bend out of shape. And by doing that, it stops further replication of the cells because it stops repair. Essentially, what happens here is that we can't repair that DNA, and so the cell has no option but essentially to kind of commit suicide, something called apoptosis. It goes through programmed cell death. It can't repair itself, and therefore it has to sort of die off. And so cisplatin bonds to the nitrogen atoms on 2 adjacent guanine bases, those G bases, on a single strand of DNA. It's something that we call an intra-strand link between the same strand. And so you can see these sort of cartoons on the side demonstrating how that works. As the nitrogen atom on the guanine bases have lone pairs, and so these lone pairs form coordinate bonds to the platinum. So how it works is we start with our cisplatin, our 2 chloro groups, our 2 amino groups. This ends up being hydrolyzed in the body, so we get diffusion out of the bloodstream and into the cells. We then get our hydrolysis reaction. So what that means is that we replace our chloro groups with water, and then these water groups themselves are then replaced by nitrogen atoms on the bases from the guanine bases in DNA. And remember that nitrogen, this is a favourable reaction because nitrogen is a better ligand than oxygen. Nitrogen is less electronegative than oxygen, more likely to be willing to give its lone pair up and act in a coordinate bond. And so from a kind of inorganic point of view, this is what we call a ligand substitution reaction. Now, cisplatin also has some side effects. It doesn't just bind to the cancerous DNA, but it also binds to DNA in normal, healthy cells too. Now, it's not so much of a problem. The reason why it works is because cancer cells are replicating much, much more quickly than healthy cells are, and so the effect of the drug is much greater on cancerous cells than it is on healthy cells. But it still affects healthy cells as well. So it's why often chemotherapy patients end up losing their hair, because the types of cells that grow quickly anyway, such as hair follicles, are affected to a greater extent as well. Another thing to say is that you can also get resistance to cisplatin. So where in the middle sort of cartoon here on this slide, we've seen that once the cisplatin binds, it distorts the DNA. And the idea of how it works is that by distorting the DNA now can't be repaired, and so it has to go through that sort of programmed cell death, apoptosis. But there is another idea where we can actually, repair can happen. We develop resistance. The body actually develops a way to repair this DNA, and then resistance happens. And so by repairing it, we can kind of get rid of the cisplatin from the cell, and the cell is still cancerous and still alive. And so it's possible to build up a resistance to cisplatin, in which case you then need to try other forms of chemotherapy. So time to have a go at some questions. If you want to have a go, pause the video and the answers will be on the next slide. So we've got some questions to deal with components of nucleotide, the bases that pair together, and the kind of forces that allow substrates to bind to enzymes, and how anti-cancer drugs work. So the first question was to say what the three components of a nucleotide were. And the three components are very simply a phosphate group, a pentose sugar, simply just a sugar, pentose meaning it's 5 carbons long, or a 5-membered ring, really it's 4 carbons with an oxygen in there, and a nitrogen base, one of those CG or A and T bases. Then which bases pair together in DNA? Remember, it's G and C go together and A and T go together. We have our sequence, and then if we're going to give the complementary sequence, we just have to give the kind of opposite, knowing that A and T bind together. So if we see an A, we write a T. If you see a T, you write an A. And also the same for G and C. If you see a G, you write a C. If you see a C, you write a G. Then we're asked which base pair is the most stable. It's remembering that C and G has 3 hydrogen bonds between them, whereas A and T only has 2. And so because we have 3 versus 2, the intermolecular forces are stronger for C and G than they are for A and T pairs. Question 2 was to ask the forces that allow a substrate to bind to an enzyme, and the forces are these non-covalent intermolecular forces. So it's stuff like hydrogen bonding, dipole-dipole forces, van der Waals forces, sometimes even ionic bonds. But the key thing being it's a non-covalent interaction. And then finally, was asking why transplatin was inactive as an anti-cancer drug. So transplatin has the structure shown at the bottom here. We still have our 2 NH3 groups, we still have our 2 Cl groups, but now these are trans to one another, 180 degrees opposite each other. And so if we were to use this as an anti-cancer drug, we'd put it into the body, and the hydrolysis reaction that we saw would occur as well, the Cl groups being replaced for water groups. The problem is now, remember, that in order for it to work, it has to bind to adjacent guanine bases that are next to each other in single strands of DNA. Now for cisplatin it can do, because the water groups that are going to get replaced in the ligand substitution reaction are 90 degrees away from each other. They have the correct kind of spatial distance away from each other to be able to bind to adjacent guanine bases. Transplatin, it can make one bond to the guanine using one water molecule displacement, but the other water molecule is too far away. It simply cannot actually kind of constrain itself to be able to bind to that other guanine base. It's just too far away. And so that's why it's inactive. It doesn't have that such radical distortion of the DNA, it doesn't bend it so much out of shape, and therefore the repair can happen. Thank you for watching this video. You can find more videos like this on my YouTube channel. If you found this video helpful, then please consider leaving a review on one of my tuition websites. I offer tuition online for chemistry. If you're interested, you can check out the tuition pages too. And if you need any more help with your chemistry, feel free to drop me an email. I'm always happy to help.